Good day, everyone. It's my pleasure to give this lecture. Skeletal muscles. The skeletal muscles are very unique tissues in our body. Skeletal muscles are attached to the bones or the skin. That's because the skeletal muscles provide the force for our movements. Skeletal muscles are longest of all muscles. Our human body has three types of muscles: skeletal muscle, cardiac muscle, and small muscles. So the skeletal muscles actually are the longest ones. Skeletal muscles have striation, which is also called the straps. Skeletal muscles are voluntary muscles. That's because the skeletal muscles can be consciously controlled by the brain. Skeletal muscles can contract rapidly, get tired easily, and contract powerfully. So the keywords for the skeletal muscles are the skeletal, striation, and voluntary. Skeletal muscles provide force for our movements. For example, when we walk, we need a skeletal muscle. Skeletal muscle maintains the posture and body position. So when we stand, when we sit, we also need a skeletal muscle. Skeletal muscles surround the joints and stabilize the joints, maintain the joints movements. Skeletal muscles can generate heat as they contract. For example, in the winter, when we feel cold, we shake our body so the skeletal muscle can generate heat to maintain our body temperature. In this schema, we can see the skeletal muscles are innervated by the nerve from the brain and the spinal cord. So that's why the skeletal muscle can be consciously controlled by the brain. In this diagram, we can see this is muscle fiber. Muscle fiber is also called as the muscle cell. So the muscle fiber actually is covered by a membrane, which is called sarcolemma. We can see this round purple structure, which is called the nucleus. Whereas the muscle fibers also contains hundreds of small fibers. These small fibers are called myofibrin. Myofibrin has dark bands, which is called A bands, and light bands, which is called I bands. You can see this red structure, so they are called mitochondria. Mitochondria can provide energy for muscle contraction. Now let's zoom in the myofibrin. We can see this brown vertical strand, which is called amylase. Where the amylase actually is in the middle of this red strand. This red strand are called thick filament. Thick filament is made of the myosin. Here we can also see this blue strand. This is called a thin filament. Thin filament actually is made of actin. So the thick filament is also called A band because this is the dark bands. In the region between the two adjacent A bands, so this is called I bands. In the middle of the I bands, so we can see this vertical zigzag strand, which is called Z discs. So the region between the two adjacent Z discs, which is called sarcomere. So the region between the two adjacent I bands, which is called the H zone. Now let's zoom in the sarcomere, so we can see the thick filament actually is made of the myosin. So the myosin has a myosin head, which can attach to the thin filament. Thick filament actually linked to the Z discs with elastic fibers, which is called titin. So we have mentioned that thin filament actually is made with actin, and thick filament is made by the myosin. So the myosin actually attached to the amylase. So if we make a cross section on the sarcomere, we can see we can see the hexagonal arrangement of one filament with 6 thin filaments. Now let's talk about the sarcoplasmic reticulum and the T-tubule. T here means transverse tubule. So the T-tubule actually is formed by the protrusion of sarcolemma deep into the cell interior of the muscle fiber. So it can increase the muscle fiber's surface area to increase the efficiency of the substance exchange, and can also allow the action potential travel into the muscle fibers. So the T-tubulum actually is connected with the extracellular space. So the T-tubule actually penetrated at the AI band junction. So the T-tubule actually is located between the two terminal cisternae of sarcoplasmic reticulum. So these three structures are called a triad. So triad actually consists of the one terminal cisternae of one sarcomere, one sarcoplasmic reticulum and T-tubule, and another terminal cisternae of sarcoplasmic reticulum. So here we can see this vertical green structure. This is T-tubule. T-tubule is located in the junction of AI bands. And then we can see this blue structure, which is the sarcoplasmic reticulum. So this bulge sac actually are called cisternae. Okay, cistern, terminal cistern. So this terminal cistern of sarcoplasmic, and this is another terminal cistern of sarcoplasmic reticulum. So these three structures actually are called a triad. So now let's talk about the neuromuscular junction. So once the nerve action reach to the muscle, so the action actually will divide to many branches. So each branches will anchor to the one muscle fiber to form the neuromuscular junction, which is also called the motor end plate. So each muscle fibers can be innervated by one branch of axon by forming the neuromuscular junction. So once, in the end of the axon, so actually there is a bulge structure which is called the axon terminal, which is also called the synaptic bulb. Between the synaptic bulb and the muscle fibers, so there is the gel-filled space which is called the synaptic cleft. So in the synaptic bulb, actually, there are many small vesicles. They are called synaptic vesicles. Synaptic vesicles contains neurotransmitters, which are called acetylcholine. Whereas the sarcolemma under the synaptic bulbs actually has many folds. So they are called junctional folds. So they contain millions of acetylcholine receptors. So briefly, neuromuscular junction consists of axon terminals, synaptic clefts, and junctional folds. So in this schema, we can see this is the nerve fibers. So this is axon terminal. So this is the bulge structure, which is also called the synaptic bulb. So in the synaptic bulb, you can see there are many vesicles, so they are called the synaptic vesicles. Synaptic vesicles contains acetylcholine, so these yellow dots actually are the synaptic choline, are the acetylcholine. So this is mitochondria. You can see this green structure actually. So this is the synaptic cliffs. So we can see this folding structure of the sarcolemma, which is called junctional folds. Okay, junctional folds have millions of receptors. Those receptors are called acetylcholine receptors, so they can bind with acetylcholine. Now let's talk about the muscle contraction. Muscle contraction have these 4 events. The first event is the events at the neuromuscular junction. The second is muscle fibers excitation. The third is excitation, contraction, coupling. The last one is cross bridge cycling. So this is the events happens at the neuromuscular junction. So we talk about this detailedly with this schema. So when the action potential travel to the axon terminal, so they can reach to the synaptic bulb. So actually, they will stimulate the calcium receptor on the synaptic bulb. So the calcium channels open and allow the calcium ions rush into the synaptic bulbs. So the calcium ions will bind to the synaptic vesicle to stimulate the synaptic vesicle. So the synaptic vesicle can use energy from the mitochondria and then travel to the axon terminal membrane. And eventually they will fuse with the terminal membrane and release the acetylcholine to the synaptic cleft. The acetylcholine can travel across the synaptic cleft and reach to the junctional folds, then bind with the acetylcholine receptors. So once they bind with the receptors, so they can stimulate the receptors and then let the receptors open. Once the receptors open, so they allow the sodium ion rush into the cells and a few potassium ion rush out of the cell. However, there are more sodium ion rush into the cell to cause the sarcolemma depolarization. So this depolarization actually is called end plate potential. So this is the video for this E1. Please watch this video after this lecture. Now let's talk about the action potential. So the action potential, muscle fiber action potential. Muscle fiber action potential is also called muscle fiber excitation. So the action potential is generated by these 3 steps. So the generation of end plate potential, depolarization, and repolarization. So we have mentioned that end plate potential actually is generated by the opening of acetylcholine receptors. Then the end plate potential can travel to the adjacent sarcolemma to stimulate the sodium channel and let the sodium channel open to allow sodium ion rush into cells to generate the action potential. So this is called depolarization. And then the sodium channels closed, but the potassium channels opened to allow potassium ions rush out of the cells to decrease the membrane potential. So this is called repolarization. So do you guys still remember the end plate potential? So this end plate potential is generated from the junctional fold. So the end plate potential can then travel to the adjacent sarcolemma to stimulate this sodium channel. The sodium channel open to allow the sodium ion rush into cells to increase the membrane potential to generate the action potential. And then the action potential can actually close the sodium channel and open the potassium channel to let the potassium rush out of the cell to decrease the membrane potential. So this is called repolarization. So in this figure, we can see this is resting membrane potential. It is negative 90 millivolts. So the end plate potential actually is negative 50 millivolts. It's here. So the end plate potential can actually stimulate the sodium channels, let the sodium channels open. Then, so the huge amount of sodium ions rush into the cells to increase the membrane potential to positive 30 millivolts. So this is action potential. So the positive 30 millivolts membrane potential can close the sodium channel and open the potassium channel to allow the potassium rush out of the cell to decrease the membrane potential, so decrease to the negative 90 millivolts. So this is repolarization. Next is the excitation contraction coupling. So in this step, actually, the action potential will induce the muscle contract. So let me talk about this. So since the action potential is generated here from here, right? And then the action potential will travel to the triads and then continue run into the T tubule. So this is T tubule. So here we can see the action potential run into the T tubule. So guess what? So there are some of the calcium channel receptors located in both terminal cystine and T tubule. So these proteins can be activated by this action potential. So they will release the calcium ions stored in the sarcoplasmic reticulum to the cytosol of muscle fiber. Then the calcium can bind with the actin to cause the myofiber contraction. So this is called excitation contraction coupling. So this is the video of the excitation contraction coupling. So please watch this video after this lecture. Now let's talk about the cross bridge cycling. Okay, so for this step, we need to memorize this structure: tropomyosin, myosin head, and myosin fiber. So again, so actually tropomyosin actually blocks the binding side of the actin. Okay. However, once the calcium is released from the sarcoplasmic reticulum to the cytosol of the muscle fiber, guess what? So the calcium actually will bind with the tropomyosin and cause the tropomyosin twist to expose the binding side of actin. So once the tropomyosin twists, so the myosin actually can bind with the binding side of actin. Then we can see here myosin head attached with the ADP and phosphate. So these two substances can be released. So the myosin head can pull the actin toward to amylin. The next step. Next we can see a molecule of ATP can bond with the myosin head. So the myosin head can be detached from the thin filament. So next step, ADP is degraded to ADP and phosphate. So the energy released, so the released energy will be captured by the myosin head. So the myosin head can extend to attach to the next binding site of the actin. So this cycle will be performed again and again to let the myosin head continuously pull the thin filament towards the amylin. So this is how the muscle fiber contract. So this is the video for the crossing bridge cycle. Please watch this video after this lecture. Now let's talk about the muscle disorders. So we have 6 types of muscle disorders. The disease affect neuromuscular junction, the denervations of muscles, muscle dystrophies, muscle hypertrophy, metabolic myopathies, and muscle paralysis. So in this lecture, we talk about this one and this one. So the first disease we talk is myasthenia gravis. Myo here means muscle. Asthenia is Latin word, means weakness. Gravis is also a Latin word, means serious. So again, this term means serious muscle weakness. So this is autoimmune disease. Autoimmune means our human body generate the antibodies attacked our own organs or tissues. So the antibodies actually destroy the acetylcholine receptors on the junctional folds. Okay, so the patient will have the mild or severe symptoms such as muscle weakness, fatigue. Okay, sometimes patients have severe symptoms such as respiratory paralysis. The patient can die. So do you guys still remember where the acetylcholine receptor are? Actually, they are here on the junctional fold. So we can use the radioimmunoassay to detect the acetylcholine receptor antibodies. Normally, we can get a 90% positive of the patient. Okay, we can also do the histology study, so the doctor can pick a small piece of samples and visualize the samples on the microscopy, so they can see the decreased number of subneural cliffs, okay, widening the synaptic cliffs and hyperplasia of thymus. That's because the study has shown that the tumor in the thymus actually generates acetylcholine receptor antibodies. So the treatments, we can use the neostigmine to inject a patient. Neostigmine actually is a reversible acetylcholine enzyme inhibitor. So if we inhibit the acetylcholine enzyme, so we can increase the acetylcholine concentration in the synaptic cleft. We can also use immunosuppression treatment. That means we can use the high dose of cortisone to inhibit the antibody production. We can also do a surgical removal of the thymus, which is called a thymectomy. So if we remove the thymus, so the antibody production will be decreased. The last step we can use is plasmapheresis. So we can use the plasma of healthy people to replace the plasma of the patient to decrease the antibody volume in the patient. So this treatment actually is used in the patient with severe symptoms. So the second disease we talk about is muscular dystrophy. So this term means degenerative disease, okay, cause progressive weakness of skeletal muscle. Patient may have mild or severe symptoms. Actually, this disease is induced by the genetic mutation on the dystrophin. Dystrophin is one type of glycoprotein. Glyco here means sugar. So glycoprotein means the protein has a sugar chain. So this is called glycoprotein. Dystrophin actually is a very huge molecular protein. Okay, so that's why the mutation can happen at different sites. So in this schema, we can see this is dystrophin. Okay, so this is muscle myocyte membrane, which is also called sarcolemma. Okay, so on the sarcolemma, we can see this complex. This is dystroglycan complex. Actually, the dystrophin, we can see dystrophin has two terminals. One is called the dystroglycan binding end. So this terminal actually anchor on the dystroglycan complex. Another terminal is called actin binding end. So this terminal actually anchor on the actin cytoskeleton. So that's why dystrophin can strengthen the sarcolemma to prevent the sarcolemma rupture during the myofiber contraction. Okay, that's because the myofiber contraction is very strong. It can cause the sarcolemma rupture. Okay. However, if the mutation happens, guess what? So the dystrophin will lose its dystroglycan binding site. So the dystrophin cannot link the sarcolemma with the cytoskeleton. So that's why the dystrophin cannot strengthen the sarcolemma, cannot protect the myocyte membrane from the cannot prevent myocyte membrane rupture from the muscle fiber contraction. So here, when the muscle fiber contracts, so the sarcolemma is broken. When sarcolemma is broken, guess what? The myofiber is die because everything inside of the cell will be released to the extracellular space, so the cell actually die. So once the muscle fiber die, so guess what? So they will be replaced by the fibrous or the fatty tissues. So the most common disease in the muscular dystrophy actually is Duchenne muscular dystrophy. So this is X-linked recessive disorder. That means the females carry mutant gene. However, the males actually are the patients. So the patient actually will be dying by the age of 30. Okay, some patient may have completely some patient may have the dystrophin protein absent in their muscle. So here we can see, so this is the dystrophin gene. Dystrophin gene actually is located in the X chromosome. So in the female has two dystrophin gene. However, the male only have one X chromosome. So that means male has only one dystrophin gene. So this is the Duchenne patient. Okay, so the female only have one muted gene. Okay, because this is recessive disease. Okay, so since the female has also one normal gene, so that's why the female will not have the disease. However, the male has only one X chromosome. So if the male has this one muted genes, so the male will have this male will be the patient. Okay. So this is genetic disorder. So the patient will have the symptoms by age 4. Okay, patient will have the muscle hypertrophy, muscle weakness. Patient will also have the cardiac muscle weakness, has the enlargement of the heart, which is called cardiomegaly. So patient will have a unique symptoms, which is called Goran sign. Okay, that means the child need his arm or hands to help them stand up. Okay, by age 12, the patient will lose all their legs muscle. So because the child legs so the children legs will not have so we are not provide enough force to help them to stand up, so they need the wheelchair. Okay, by age 30, so they will die. So this is Gover sign. Okay, so the patient actually cannot stand up with their leg. Okay, so they need their arm or hand push on the ground to elevate the upper trunk, and then they need their leg to elevate their lower trunk. So finally they can stand up. Okay, that's because their legs or their arms are weaknesses, so they can stand up with their legs. So they need the arms and hands to help them stand up. Thank you very much. Okay, so I take your questions.